So if you cannot find a seat, you are welcome to come to the front. We're not going to bite you. My name is Santiago Schnell. I'm the William K. Warren Foundation Dean of the College of Science. And I would like to welcome you to today's Gold Mask lecture. The Gold Mask began at the Massachusetts Institute of Technology in 2016, named for the color of the hoods worn by individuals graduating with a doctoral degree in science. The Gold Mask is also associated with Saint Albert the Great, the patron saint of saints. We at Notre Dame hosted our first Gold Mass, followed by a lecture in 2017. Why we are doing this? Notre Dame has a rich history of connecting science with the Catholic faith. The connection began as far as back as the early 1890s with John Sand, a Holy Cross priest who taught science and served as the chair of the Department of Science at Notre Dame, which is a predecessor of the College of Science. He founded a magazine called Catholic Science and Catholic Scientists, which combined with his lectures culminated in an infamous book at the time, Evolution and Dogma, published in 1896. It was controversial, ignited concerns with the Vatican, and quietly ordered it out of circulation. However, thanks to his contributions to the dialogue between science and faith, Pope Leo XIII awarded him an honorary doctoral degree in philosophy to Dr. John Sand in 1895. And apparently, this is one of the only two such degrees ever granted by the Vatican. Through the Gold Mask lecture series, we're following Father Sand's spirit of engaging in a dialogue of topics at the interface between science and religion. And today, I have the pleasure of introducing our 2023 Gold Mask speaker, Dr. Maureen Condic. Dr. Condic holds a Ph.D. in chemistry from the University of California, Berkeley, and is an associate professor of neurobiology and anatomy at the University of Utah School of Medicine. She has an annual appointment in the Department of Pediatrics. Since her appointment at the University of Utah, Dr. Condic's primary research focus has been on the early development of vertebrate embryos. Dr. Condic's current research focuses on studying human embryonic fluid properties derived from stem cells to study how those fluids can help on the differentiation of immature cardiac cell muscles. She's also interested in understanding the human stem cell potency and differentiation. In addition of her research, she teaches both graduate and medical students. Her teaching focuses primarily on embryonic development, and she has also taught human embryology at the University of Utah School of Medicine curriculum for over 20 years. The reason we have invited her is because in 2020, she published a book that is called Untangling Twinning, What Science Tells Us About the Nature of the Human Embryo. I had the privilege of reading this book a couple of years ago. It is one of the best reviews of the current literature related to human twinning, which brings state-of-the-art biological knowledge to the philosophical discussions about the human nature of individuality. And this is a very critical point because it's used by the law to do experiments with human embryos. So it's a very hot topic. She also co-authored Human Embryos and Human Beings that explore the nature of the human embryo from the scientific and philosophical perspective. Dr. Condic is an internationally renowned scholar. She's a member of the Pontifical Academy for Life and serves on the National Science Board. Those of you who don't know what the National Science Board, the member is composed, I think it's 20 individuals that compose them, and they actually help the National Science Foundation to set up policies for research funding and serve as advisor for both the Congress and the President of the United States. In today's talk, Dr. Condic will describe how, although human life begins at the sperm-egg fusion, one may resolve the questions rise when the single embryo splits into two or more genetically identical human beings. She will share the scientific evidence for when a human life begins and when the human identical twinning occurs, and will examine the philosophical issues raised by the phenomenon of human twinning. Let's give a warm welcome and thank her for coming here. Thank you.
Thank you so much. Well, I am thrilled to be here today to talk to you, the members of the University of Notre Dame, and to talk about one of my favorite topics, a topic near and dear to my heart, the human embryo. So, in particular, I'd like to focus on the phenomenon of twinning. So, human twins have long been the subject of myth and mystery. This is, of course, one of the earliest known portraits of Romulus and Remus. Oops, not my pointer to point. The famous twins who were the founders of Rome, being nursed by their wolf mother. But twinning, other than sort of in story and fable, really became an issue with the advent of in vitro fertilization in 1978. So this is a picture of the first test tube baby. And the development of IVF raised a lot of concerns, a lot of ethical concerns, not merely about twinning, but about the nature of the embryo. And in support of this practice, Clifford Grobstein, who was a faculty member at one of the California universities, I think California Davis, proposed the concept of the pre-embryo. So one can debate why Grobstein felt he needed to weigh in on this topic, but certainly it was in the economic advantage of his university, who wanted to provide IVF. It was in his own scientific advantage because he wanted to be able to work on embryos. And it was in the advantage of calming the public, trying to keep people from getting up in arms about the prospect of producing embryos in the laboratory. So Grobstein wrote this paper, published in Scientific American, called External Human Fertilization. And in it, he stated that the stages involved in IVF were not only pre-persons, but also pre-embryos. They're pre-embryos because separated cells from the early cleavage stages of eggs of non-human mammals, and most likely also of human beings, can each give rise to a whole embryo in twinning. And because the cells can be deleted from early clusters, or two clusters can be combined, and a single embryo can result. Therefore, in the cellular stages prior to implantation, a single embryo has not yet been established. So he's referring to the ability to take cells out of an embryo, to add cells into an embryo, and to split embryos into two embryos, and to combine two embryos into a single embryo. So this plasticity in early development convinced Grobstein that there was really no embryo at this point. This concept was taken up by a very influential book published by Father Norman Ford, a Salesian priest in Australia, When Did I Begin? And due to the possibility of human twinning, Ford argues in his book in 1988, an early embryonic human cells cannot constitute an actual human individual. Instead, they would have the potency to form one or more human individuals. It seems that the biological evidence leads to the philosophical conclusion that a human individual, our youngest neighbor and member of the human community, begins at the primitive streak stage, which I'll talk about a little bit later, and not prior to it. So both of these arguments were taken up by a number of different countries. For example, England, in the Warnock Report, used this pre-embryo argument to establish the 14-day rule, the point at which twinning was no longer possible and an individual arose. And this became the basis of legislation on embryonic research throughout the world, including in the United States. I would argue, however, that the timing of twinning matters. So if you start with something that's unambiguously a human person, a mature individual, and if it were possible to twin this person, twinning after the nature of the entity as a human being has been clearly established really does not call into question the identity of the original human being. So if I were to take a skin cell out of this lady's hand and use it to clone an identical twin of her, she'd be much younger, but she would be an identical twin. And yet nobody would say, well, that means that this person wasn't an individual yet. The problem with twinning at early stages of development, so this is a one-celled human embryo, if it divides into two cells, and if each of those cells can go on to make a separate mature individual, then for many people this raises doubt about the nature of the original cell. Precisely because in their minds, the identity of this thing as a human being has not been determined. So the problem of twinning, put simply, is this. If a zygote, or a one-celled embryo, divides to produce two identical totipotent cells, both of which, if separated, can develop into a mature individual. So we make two cells. If we separate those cells, we now have two individual cells, each of which has the ability to go on and produce a mature individual. Then how can that original cell be a single human? So these are actually, believe it or not, different individuals that just look extremely similar, adult identical twins. So addressing human twinning, we're going to start with the biological issues by asking when does the human zygote form and what happens in early human development. How long does totipotency persist? And when does twinning actually happen? And then we're going to move on to the philosophical issues, the question of virtual humans, or what's the different meanings of the term potency as it's applied to human development. Does the original individual who produces the twins survive or die? Who are the parents of the twins? And the question of identity in form and matter versus ontological identity. So we'll unpack all of these philosophical issues in greater detail as they come up. What I'd like to start with is just a few slides on early human development. So in about 4 or 5 slides, I'm going to give you what it takes me a semester to do for the medical students. So consider yourself blessed. We're not going to go into that much detail. What you're going to see is a movie. This is the first 5 days of human development. This is taken from a fertility clinic. So this bigger circle, bright circle, is a culture well. This grainy circle in the middle is the one-celled human embryo, or zygote, shortly after sperm-egg fusion. And it's surrounded by a clear gelatinous layer that doesn't contain any cells. That's called the zona pellucida, or the clear zone. So this is sort of a jelly coating that protects the egg when it's ovulated and protects the embryo during the first 4 or 5 days of development. Okay. So what you're going to see in this movie is that the early embryo enters into a rapid period of cell division. So it divides in half without growing in size. And these early cells that are produced, these smaller cells, are known as blastomeres. After about 3 rounds of cell division, we reach what's called the morula stage, because it looks kind of like a raspberry. And at this stage, an important change happens in those blastomeres that are produced. They change their properties so that they can adhere more tightly together. And as a consequence of that tight adhesion between the cells, the embryo can now transport fluid to the inside, and the fluid can't escape. So you see this whole thing blowing up, kind of like an inflating balloon. And after about 5 days, it reaches the blastocyst stage, which is what you're looking at here. So you can think of the blastocyst as kind of a fluid-filled soccer ball. The cells on the outside, the black and white hexagons of the soccer ball, those are cells known as trophectoderm cells, and they're going to largely go on to produce the placenta of the embryo. And a smaller group of cells on the inside, known as the inner cell mass, or the ICM, about 8 or 10 cells at this stage, is going to produce all the cells you're carrying around in your body today. So the first division of labor happens at the blastocyst stage. All of the events I just described take place in the woman's reproductive tract. So this is a cartoon of sort of the upper portion of the uterus and the ovary. Mature eggs are ovulated out into a structure known as the fallopian tube. And all the events we just talked about happen there. So sperm-egg fusion, the production of the morula, compaction, the production of the blastocyst. And then at about 6 to 7 days, we get implantation into the uterus. Once implantation has occurred, then a number of important things happen within that developing embryo. The inner cell mass cells here will produce 2 different layers. So this is now the bilayered embryo stage. And then at about 14 days, that event that I mentioned, the primitive streak formation, happens. And what that means is that cells in this upper layer are going to migrate towards the midline of the embryo. And when they hit the midline, they're going to dive deeper in and change that 2-layered embryo into a 3-layered embryo by migrating out in between the 2 layers. So that primitive streak, that midline structure that the cells will migrate towards, is the earliest marker of where the nervous system is going to form. And it's also the stage of development, about 14 days, after which identical twinning can no longer occur. So if twinning does happen, which it very, very rarely does, you're going to have conjoined twins, but not separate individuals. And even then, it can only maybe happen for 1 more day. So at the end of the 14th day, we're done with the capability of twinning. So what happens after that? These are pictures of actual embryos taken from the Carnegie collection. That one-celled embryo or zygote we talked about is this little dot over to day 1. It's blown up here so you can actually see it. And these pictures represent the first 8 weeks of human development, so the so-called embryonic period. The embryonic period, this first 8 weeks, is the time over which it takes you to go from a single cell all the way up to a fully formed, albeit very small, only 5 millimeters here, human body form. So embryogenesis is the time that all the tissues, structures, systems, organs, and relationships in the embryo are formed. After that, the remainder of prenatal development is the fetal period, so fetogenesis. And during that time, the fetus is going to grow much larger in size. The tissues are going to mature chemically and physiologically, but you're not going to make anything new. So during those periods, starting the start of the fetal period, the fetus is going to grow enormously in size. So here by 10 weeks, you can see fingernails and eyelashes. 14 weeks, proportionally much, much larger. And by 20 weeks, a fetus is about 6.5 inches long. It weighs about 3/4 of a pound. It's moving. It's reacting to sound, to touch, to pain. It can learn things from its experiences. And if given appropriate intensive neonatal care, 23% of the infants born at this stage will survive, and the majority of those without long-term deficits. So it's pretty impressive that at such a tender age, you have such remarkable abilities. But although those pictures and that description tell you what happens in early human development, it doesn't answer our question very, very well. When does human life begin? And to answer that question, we immediately run into what I call the continuum problem, which is that because development progresses continuously from 1-cell stage up through birth and beyond, many people are going to set pretty much arbitrary points for when life begins and for when human beings have rights. Lots of people like blastocyst formation, or that first division of labor in the cells within the embryo, at about 4 days after sperm-egg fusion. Some people like implantation, or the point at which the embryo first establishes a relationship with the mom. Some people like formation of the primitive streak, or the initiation of the nervous system, or neural development. But you can go later than that, kind of analogously to brain death. A lot of people advocate this very fuzzy concept of brain life, or the point at which we have the onset of mature patterns of neural activity in the brain, something probably around maybe 5 months of development. But you can go even later. Peter Singer at Princeton is famous for having proposed self-awareness as the relevant point at which human value and human life begin, an event that he pegs at something around 28 days after birth. And as a developmental neuroscientist, my personal favorite, given the very long time over which the brain develops, would be this one, brain maturation, something that doesn't even happen until about 25 years post-birth. So, you know, a number of you are not even humans yet. We could send you back to the human parts factory, right? Okay, but what I hope these examples can tell you is that choosing among these definitions really is not a matter of science. It's just a matter of utility or taste, or I would also add power. So how then does science address this question? Well, thankfully for us, well, not so thankfully, we have to ask 2 related questions to answer this one question. First, we have to ask, when is a new cell type produced from the interaction of sperm and egg? So long as the gametes, the sperm cells and the egg cells persist, we can't have anything new. And secondly, what's the difference between a human cell and a human being? So, truly, thankfully for us, the first question is a very simple question to answer. Scientists use two very easy, straightforward criteria to determine when a new cell type forms. Changes in cell type are detected by changes in material composition, what a cell's made out of, most typically due to changes in gene utilization. So we turn a gene on, we turn it off, we can change what molecules are present within the cell. Or changes in cell behavior, and very often those two things go together. You give cells a new toolkit, they can do different things. I can assure you these two criteria are used throughout the scientific enterprise to determine when a new cell arises either in an embryo or as a consequence of a manipulation in the laboratory. Logic, of course, also requires us that if a new cell arises from the interaction of two or more pre-existing cells, those cells have to go away before we can have a new cell. So, based on this criteria, on these criteria, is the product of sperm-egg fusion a new cell, or is it just a modified gamete? People often refer to the product of sperm-egg fusion as a fertilized egg. So is that a scientific term? Is that an accurate way to describe what happens, what you get after sperm and egg fuse? Well, I hope to convince you that fertilized egg is a completely nonsensical term, and there is no such object, because a new cell unambiguously forms at the interaction of sperm and egg. So this is an electron micrograph showing a much larger pink egg cell and a much smaller blue sperm cell that are fusing. And upon sperm-egg binding, so upon the surfaces of these two cells coming together and the molecules on those surfaces interacting with each other in a stable manner, the plasma membranes, or the outer lipid layers of these two cells, will fuse in a very well-studied and very rapid process that takes about 250 milliseconds to complete. So it's a quarter of a second, you go from two cells to one cell. Once that membrane fusion has happened, all of the lipid and protein that used to be uniquely present on the surface of the sperm is now free to diffuse into the surface of this new cell and change its molecular composition. In this process, the sperm itself ruptures, it ejects its nucleus and all of its cellular contents. And so clearly the sperm as a separate cell ceases to exist in this instant, well-defined instant of sperm-egg fusion. What about the egg? Well, following the fusion and destruction of the sperm, the resulting cell has a new composition that's distinct from that of an egg cell. We've already talked about the incorporation of lipid and protein from the sperm's surface into the surface of this new cell, but the changes are much more extensive than that. The cell now has a new genetic composition, with half of its DNA being derived from mom in pink and half of it from dad in blue. But perhaps more compellingly, this newly formed cell radically changes its behavior and acts in a very un-egg-like manner. So the entire raison d'etre of being an egg, the entire function of a gamete is to find a gamete of the opposite sex and fuse to it. That's what they're designed to do. That's what their function in development is about, their function in physiology. And yet, within 1 to 3 seconds of sperm-egg membrane fusion, this new cell will initiate a molecular cascade that over the next 30 minutes is going to result in the deposition of enzymes into the space between that clear protein jelly coat, the zona pellucida, and the surface of the cell. And the function of these enzymes is to antagonize the very function of the egg cell itself. It's to destroy all of the remaining sperm binding sites. So the entire thing an egg wants to do is fuse to a sperm. The very first act of this new cell is to enter into a trajectory that is incompatible with the function of the gametes from which it's derived. So the egg, as a haploid cell designed to fuse to a sperm, also obviously ceases to exist in this instant of sperm-egg fusion. And a new cell with unique material composition and unique behavior and unique genetics is formed. So what is this new cell? Well, its scientific name is, of course, the zygote, or one-celled embryo. But looking at this cell doesn't really give us a lot of insight into its nature. So is the zygote a new individual human being, or is it perhaps some other kind of a cell, a pre-embryo, as Dr. Grobstein elected to call it. And I think the difficulty we have looking, even I as an embryologist, looking at this picture, is that at early stages of development, recognizable human form and function are not evident. So how do we know that a zygote or a morula or a blastocyst is a human being and not merely, a human cell or a ball of human cells or a pre-embryo on its way to becoming a human being? Well, to answer that question, we need to ask the question of how do cells behave compared to how embryos behave. So this is a picture of what a typical cell in laboratory culture would look like. And I can assure you that the vast majority of cells in your body and most cells in the laboratory don't do a lot of anything. Most cells won't even divide. However, when cells do divide, they act like little Xerox machines. So they produce identical copies of themselves. Some rare cell types are able to behave a little bit differently. They can generate progeny that have different properties from themselves in different culture environments. So in this example, if you culture a stem cell in either a low level of oxygen or a high level of oxygen in the atmosphere, you can get cells with different properties. So the different colors of the cells you're seeing here are simply colors that identify different molecular properties. So different colored cells are different kinds of cells. And yet, in all cases, cells are produced randomly with no global integration, coordination, or patterning. So all cells can do is produce tumors, even when they can produce tumors that contain many different types of cells. In contrast, how do embryos behave? Well, in an appropriately supportive environment, the embryo undergoes a self-directed, organized pattern of development to generate a fetus. It functions not as a cell or as a group of cells, but rather as an organism. So what's an organism? Merriam-Webster gives us two convenient definitions. One, a complex structure of interdependent and subordinate elements whose relations and properties are largely determined by their function in the whole. Or two, an individual constituted to carry on the activities of life by organs separate in function, but mutually dependent, a living being. So both of these definitions kind of stress what to me is the essential feature of an organism. An organism is made out of parts. Those parts do different things, but the function of those parts doesn't make any sense at all, except in the context of the thing as a whole. So there's no reason for a kidney to exist. Kidneys do not wander around on the street. They do not reproduce. They don't form societies or go to college, because the only function of a kidney is to function in the context of a larger entity to which that function contributes. I would also note that a living being of the type Homo sapiens would be a living human being. I often hear people on the other side of this discussion say, oh, well, human being isn't a scientific term. Scientists don't use that term, and that's, of course, nonsense, because human being is simply a synonym for human organism. Okay, so those are good definitions, but they're kind of vague. We can go to the opposite extreme. There's a paper by Bernard Rosenbach that was published in 2016 that goes on for about 60 pages and lists 10 very discrete and complicated characteristics of an organism. I think that's a little bit too much in the opposite direction. So I've tried to summarize those, or to cluster those 10 characteristics into 4 classes. One of which is development, or the ability of an organism to undergo a characteristic, to grow towards a characteristic mature state that functions as an integrated whole. Autonomy, or the self-directed activities of the organism internal to itself, including the ability to compensate for any perturbations in those internal activities, such as the ability to repair injury. Adaptation, similar to autonomy, except the ability of an organism to respond to changes external to itself, to changes in the environment so that it maintains its internal homeostasis. And integration, or the coordinated function of parts to support the function of the entity as a whole. Now, I would note these are discrete, observable biological traits. They're not mystical characteristics. They're not matters of personal opinion. They're not matters of divine revelation. They either exist or they don't exist, and they distinguish pretty clearly between an organism and a cell. So, when does organismal function begin? I think everybody would agree this is an organism. It's an entity comprised of parts, separate in function, but contributing to the health and survival of the entity as a whole. I would say even most of our interlocutors would probably call this an organism. It has parts, systems, organs that function to support the entity as a whole. But is this an organism? I have a friend who's an attorney, and every time he sees this picture, he always goes, nah, it's not an organism, that's a bear aspirin. And I have to admit, it does look a little bit like a bear aspirin, okay? But I'm hoping to convince you this bear aspirin from the one cell stage forward is unambiguously an organism. Not because I say so, but because it exhibits all the characteristics we've just defined. For example, growth towards a characteristic mature state. Well, development is the entire business of an embryo. It enters into it within 1 to 2 seconds, and it continues straightforward, does not pass go, does not collect $200, does not do anything until it produces a little small characteristic form. What about autonomy, or the self-directed repair of injury, such that the function of the embryo is preserved? Well, embryos are actually pretty good at this too. I'll give you just one example, that of pre-implantation genetic diagnosis. So what you're seeing in this image is an embryo at about the 8-cell stage. It's being immobilized against a smooth pipette here by suction, and then a sharper and smaller pipette has been inserted to remove or biopsy one cell out of this embryo. So this is typically done in fertility clinics if there's some risk that the embryo might be afflicted with a genetic condition incompatible with normal development. So typically what's done is they remove a single cell from an 8-cell embryo, and then they analyze the genetics of that cell. The embryo, in the meantime, is allowed to mature up to about the blastocyst stage. And if it passes the test, if the analyzed cell proves that the genetics of the embryo is normal, then the embryo can be transferred to the uterus. If not, then it's disposed of as medical waste. Shockingly, embryos can regenerate. They can recover from this catastrophic injury. I mean, to lose an eighth of your body at a very tender age, I mean, that's a pretty impressive thing. Most of us would not be able to recoup from an injury of that type, such that there have been thousands of live births from such biopsied embryos worldwide. What about adaptation, or the healthy adjustment to changing environmental circumstances? Well, again, embryos are really good at this, and I'll, again, give you just a single example, that of ectopic pregnancy. So, as many of you, I'm sure, are familiar, most ectopic pregnancies do not progress to completion, largely because embryos, when they're not implanted in the uterus, typically can't develop a sufficient placental circulation with mom to support their survival. However, there are cases in which embryos do survive, such as this one in 2003, where this little baby, Nahalanisi here, developed entirely to term, healthy 9-month infant, completely inside her mother's liver. So how could that even happen? I mean, well, we talked about how early development goes, and so she was formed in the fallopian tube, and she took a wrong turn. Instead of going down to the uterus, she ended up in her mother's abdominal cavity. And she was sufficiently lucky to attach to the liver, which is a very soft organ and also highly vascularized. So because of that, she was able to establish a normal placental circulation with the mom's hepatic vasculature. And at 9 months, I can't even say did a C-section, because she was never in the uterus. They just cut open the mom's abdomen and cut a little slit in the liver and popped the baby out. She was perfectly healthy. So if a baby can develop to term inside her mother's liver, I'd say that's pretty good testimony to the adaptive capability of embryos. So what about integration, or the coordinated function of the parts to support the entity as a whole? Well, again, embryos are really good at this. And I wrote a review, if anyone wants to get into the weeds on the details, summarizing hundreds of papers and looking at events that can really be only understood as parts of a developmental sequence, as coordinated activities that are designed to work together to produce natural patterns of development. And it's completely unambiguous that sperm-egg fusion sets off a molecular cascade that controls events taking place hours, days, and even weeks later in life. And that none of these events are required for the cells to be healthy. They're not cellular events. They only make sense as part of a developmental sequence that's produced by an organism. And while some scientists may dislike or deny this conclusion, it's supported by hundreds of peer-reviewed publications, and it's entirely uncontested in the literature. I've given this talk dozens of times, and I've asked, sometimes to quite hostile audiences, and I've always asked people, hit me with your best shot. Send me a paper that provides any evidence whatsoever that this conclusion is not true. And while I say, well, you can find many scientists who will say we don't know, or nobody agrees, or there's not a consensus, I have yet to be hit with anybody's shot because there just is no literature out there. So embryos are unambiguously organisms. And what can we conclude then from the facts? Based on universally accepted criteria, new cell, the zygote with unique composition, genes, and behavior comes into existence at this instant of sperm-egg fusion. It exhibits all the characteristics of a full but immature organism. And therefore, human life commences with the onset of organismal function at sperm-egg fusion, or a human zygote is a human being. And this is an objective, evidence-based definition of when life begins. Which, of course, brings us then to the concept of twinning. If we have human life starting at that zygote stage, the little bare aspirin, does the fact that that bare aspirin can split into two and generate two individual human beings challenge this conclusion? Grobstein certainly thought that it did, and so did Father Ford and many others. I'm always perplexed by this, because as a biologist, I think we're all familiar, or I'm certainly familiar, and I can help you be familiar with many examples of animal splitting. This is the most well-studied of many of them. This is a planarian organism. It's a kind of worm, a flatworm. And many, many, many decades of experiments have shown that you can split a planarian into little parts. A single worm can be divided into 256 pieces, and each of them will regenerate another mature worm. So this does not mean that there are 256 individuals resident inside of this free-living flatworm, or that the flatworm itself is not an organism. It means that animals can reproduce asexually by splitting. And we, as humans, have that capability up into about 14 days. Okay. We also know, just like a planarian, this is an organism. And if that organism divides into two cells and they're separated, just like the planarian, even if twinning is happening at the two-cell stage, twins that arise from a single organismal entity that's split does not call the nature of that original entity into question. I would also ask, when does twinning actually occur? So twinning at the two-cell stage is highly unlikely. And this is based on a couple of lines of evidence. First, people watch early human development in vitro fertilization clinics all the time. And with one author stating that despite the very high incidence of monozygotic twinning or identical twinning in IVF procedures, twinning in the first 3 days of life has never been reported, with one author stating, we've never observed an embryo splitting in half before the blastocyst stage on its own initiative in over 15 years of laboratory experience. The reason for this is also supported by a lot of, the reason that it's probably never been observed, but also the argument against early splitting of embryos is a lot of experimental evidence that indicates early embryos are very, very sticky. Those early blastomeres that are produced from the cleavage of the zygote readily adhere to each other. And we know this from a number of classical experiments, including if you take animal embryos at the 16-cell stage, shown here, you dissociate them enzymatically into a single cell suspension, and then you just clump them all back together. What will happen is those cells will tightly adhere. They're going to resort out into their original positions, and development will carry on uninterrupted. So even if you separate the cells, they have a strong tendency to try to glue themselves back together. Similarly, embryo fusion argues against embryos splitting at early stages. If you take two embryos, and a blue one and an orange one as depicted here, and you fuse them together into a single cluster, they will rapidly integrate and produce what's called a chimeric embryo, or an embryo produced of cell types, each of which has a different genetic composition. So you can do this with even different species of embryos. These are some animals that were produced in 1984 by fusion of a goat and a sheep embryo in the laboratory and then transferred to a surrogate mom. So these are unnatural creatures called geeps. They have 50% goat cells and 50% sheep cells. But clearly, they've developed as an organism in a coordinated manner due to the strong tendency of these early blastomeres to participate in a single developmental trajectory. We can do this in cooler and more photogenic ways with mice. These are chimeric mice that were made by contributions of stem cells, all of which have been colored to be identified or traceable within the embryo. Plus, we've directly tested this idea. In this experiment, the scientists created a single zona pellucida that contained 2 embryos. So you can see each of these cells has 2 nuclei. So these are 2 one-celled zygotes surrounded by a single zona pellucida. They made about 180 of these, and in all cases, 100% of those 2 embryos fused to each other and went on to produce a single embryo comprised of cells derived from each of those original 2 embryos. So it's highly unlikely that when the embryo is still in the zona pellucida, that twinning can actually happen, that you could separate those cells sufficiently, overcome their natural tendency to adhere together and participate in a coordinated pattern of development in order to get 2 separate individuals. So here's an alternative view of when twinning occurs. So you remember that zona pellucida, right? That jelly coat that surrounds the embryo. Well, the cells of the trophectoderm are the cells that are actually outer cells, the black and white hexagons of the soccer ball, are actually what adhere the embryo to the uterine lining. And so in order for that adhesion to happen and implantation to occur, you have to escape from this zona pellucida. And that happens in a process that's illustrated here called hatching. So yes, all of us hatch just like a chicken. I'm going to show you a little movie of hatching. So here you can see the embryo inflating, and it's going to secrete enzymes that will break a little hole in that zona pellucida, and then the whole thing is just going to squeeze out. Isn't that shocking? Who would have thought we were that rubbery at that age? So this is definitely a point at which twinning could occur. And in fact, twinning has been observed in fertility clinics at this stage. As that embryo squeezes out, there have been 2 published reports of the embryo breaking into 2, each of them having a little bit of zona pellucida, and, a little bit of ICM, and going on to produce identical twins when transferred. So are we done yet? Do we have any other credible concerns remaining? The biological facts indicate that a human organism forms immediately upon sperm-egg fusion, with exhibiting all the characteristics of a full and complete, albeit immature, human. And twinning is quite unlikely to happen at the 2-cell stage. So if it's happening at the blastocyst stage, we already have now 5 or 6 days of a developmental trajectory that identifies this thing as a single individual. And if it's going to split in 2 at that point, I think we're back in a more intuitively planarian-like situation. But are there any additional concerns? So here's where I have to make the philosophers in the audience happy. So this is the book that I wrote on twinning. And if you look at chapter 5, I go through a number of concerns that have been raised by philosophers. Virtual twinning, or virtual humans, the death or non-death of the original embryo, who are the parents, and this question of identity and form of matter, or ontological identity of the twins. So we're going to start with this one, virtual humans. So Dancil and Ford, the author of When Did I Begin, raised this question in this form. So if the potential to form a human person is the criteria for being a human being now, then before the loss of that potential, or that totipotency, quote, every single cell of the zygote of the morula and of the blastula is a human person. And this to Dancil seemed just absurd. Ford agreed, and he attempted to resolve Dancil's problem by holding that While a zygote has a, quote, natural active potential to become a mature person, it's, quote, an individual but not a person. And thus, twinning places the non-human individual, replaces this non-human individual, perhaps the pre-embryo, with two human persons at some later stage in development. So we need to modify both of these concerns a little bit in light of the evidence I've just presented to you. So substantial evidence indicates that totipotency is unlikely to be preserved beyond the 2-cell stage, if even then. I actually did not present that evidence today, but you can trust me on this one. Therefore, in most cases, only one developmental trajectory is initiated, and in the worst case, only 2. So it's not the case that we have hundreds or dozens of cells, each of which is a human person. And whether twinning occurs at the 2-cell stage or later, a single developing organism is split. And Donseal and Ford would have to presumably maintain that ensoulment is therefore delayed for all embryos until the point at which identical twinning is no longer possible. Yet this requires us to view a human entity that's clearly undergoing a human organismal trajectory of development as something other than a human being. And I have no idea what that thing could be, except a made-up thing, a pre-embryo. Okay, the central issue of both Donseal and Ford's concern is this one, is potency. So if a zygote is a person, in virtue of the fact that it has the potential to develop into a mature human, then all the cells with this potential would presumably be equally persons. A zygote that has the potential to be 2 persons cannot simultaneously be a single person. So how does this problem arise? Why is this such a confusing way of thinking? It's confusing because the term potency has several meanings. And I think authors like Donseal and Ford are using the term in an imprecise sort of way. So what would be perhaps a more precise way? Well, I'm going to go back to Aristotle. He considers potency along 2 distinct axes, active and passive, and proximate and remote. So let's start with active and passive. An active potency is the power to act in a specific manner that's intrinsic to the thing itself. So I have the potency to speech, even if I'm silent, which no one has ever observed except when I'm asleep. A passive potency, in contrast, is the power to be acted upon in a specific manner. So wood, for example, has the potency to be fashioned into a chair by the action of a carpenter. Water does not. And neither has the active potency to become a chair on its own. Okay, what about proximate and remote? So a proximate potency is the power to act or to be acted upon. So it can be either active or passive. But to be proximate, it has to be a potency, a power that is immediately exercisable. So I have the proximate potency to speech, even if I'm currently silent. A remote potency is the power to either act or to be acted upon in a specific manner that requires some intervening event to occur before it can be exercised. So an infant has the remote potency to speech, but it must learn how to do it first. And a sapling has the remote potency to be acted upon by a carpenter and turned into a chair, but it has to mature to the point of having wood. So a little green sapling cannot be turned into a chair, even though it could be acted upon in that way if you just let it mature. Okay, so let's apply this to the idea of a development. So a zygote has the proximate or intrinsic, or the immediately exercisable active potency. So the proximate active potency to be a human being, and therefore is a human being now because it's actively exercising an intrinsic power of maturation and development. So all on its own, with no intervening events, it goes on to do something that is only a characteristic of an organism. In contrast, early blastomeres have the remote active potency to be a human being, which means that blastomeres are parts of a single human being now. They're interacting with each other and participating in a single trajectory of development, but they can become an independent human being if an intervening event occurs. And so the intervening event would be the isolation of the blastomere. So if you do this, and people have done this experimentally. You kill off one of the first two blastomeres. Yeah, it can go on, but that's not an active. It's an active potency, but a remote one, not approximate active potency. So the remote potency of the blastomeres means that there's a single unified human individual prior to twinning, and that the blastomere has to cease to be a part to actualize its remote potential to be a whole. So they must undergo a substantial change. So what do we conclude about virtual humans? The proximate active potential to develop into a mature human is the criteria for being a human person now. This potential exists in a zygote, but not in totipotent blastomeres when they actually exist. And upon splitting or twinning, the remote active potential of the blastomere parts to independently develop as a mature human person becomes approximate potential. So there's a substantial change in the nature of those cells. And at that point, two human persons exist. Okay, let's move on to a somewhat easier one to think about. Does the original individual die or does it persist? So Ford and Anscombe both objected that twinning had to be important because there was no corpse. Ford states, it's impossible to admit that the original zygote and human being individual cease to exist when, without dying and without a dead cell remaining, it gives asexual origin to an identical twin offspring. And the same objection is raised by Anscombe. She says, but what has become of the human that both of them, the twins, were once identical with? Has he or it simply ceased to exist? As we might say, the parent amoeba ceases to exist upon splitting? Well, my answer to both of them is, well, yeah, that's what happens, because you don't need a corpse in order for something to cease to exist. So, for example, in mitosis or cell division, one cell will divide to produce two cells without generating a corpse and without any death occurring. So we have one cell, now we have two cells. And this is completely intuitive. There's no requirement that the original zygote die or that it leave behind mortal remains. Sperm and egg can fuse to produce a zygote without the death of either of the gametes. They simply no longer exist. So you can have a change in the nature of something, and that substantial change does not require death, merely a ceasing to be. So the matter that used to be part of sperm and egg or part of this single cell before it divided in this little video persists, and it receives a new form. Yet there must be evidence for the new form, so a change in its composition or behavior. And is there evidence that an embryo ceases to be after splitting? So we're going to watch a movie of what happens when you split an embryo. So in this experiment, embryos were split at the late morial stage, with the half embryos on the left-hand side and the whole embryo siblings on the right-hand side. And the half embryos and the intact siblings were simply allowed to be cultured in parallel. And this is what happens. They both undergo compaction. They inflate as little blastocysts. They do it absolutely in synchrony, with the half embryos simply being half the size. So this experiment tells us that there's no death here. There's a continuation of the original developmental trajectory. The half embryos do not restart from some sort of totipotent state, but they simply regenerate and develop in synchrony with their unsplit siblings, which means there's no evidence for a ceasing to be. If you start with the whole embryo, you cut it in half, and we'll just, for simplicity, consider only one half we destroy, one half we allow to persist. The persisting half immediately continues as a unified whole. So it repairs that injury and goes on with development without even a pause. And therefore, it's reasonable to think of the surviving half as an injured embryo. We start with a whole embryo, we have an injured embryo, and we have a healed embryo. And logically, the ontological status of this half does not depend on the as of yet undetermined status of the second half that we threw away. So what about the second half? So if an embryo is split in half and both halves survive and regenerate, there's still no evidence that the initial embryo ceases to be. Suggesting that one of the twins is ontologically identical is the same individual as the original individual. And the other one is a newly generated individual. And our inability to tell which twin is the original and which twin is the new individual is an epistemological problem. So it's a problem of knowledge. It's not a metaphysical problem or a problem of what something actually is. Okay, who are the parents of the twins? This one's probably going to bring me the most grief on my head, but I'll go out on a limb and make the argument anyway. So Wallace raises some who's your daddy objection. So twinning would be, quote, an incidence of asexual generation, the production of one individual from another of the species, a type of generation that is found in lower forms, but is not proper to humans. So is asexual reproduction proper to humans? It's not familiar. We don't do it in mature stages. We're not planaria. You can't cut us into 256 pieces as an adult and make a whole bunch of us. Boy, that would be helpful. I could have one person to do my laundry, one person to stay nice and fit, one person to like read novels and eat chocolate. You know, I would love that. Okay, but the answer is we don't do it except during the first 14 days of life. And it is natural for us. It's a perfectly proper form of human reproduction. I think underlying Wallace's objection is the question of parenting and the relationship of the twins to each other. In Wallace's view, if twinning initiates with an actual human person, the zygote, then the original person or zygote is both the brother or the sister or the father or the mother of the twin that's generated, and this appears to be unseemly. So how do we get out of this? I think this concern can be addressed by drawing a distinction between random and intentional acts. And this requires us to unpack the meaning of the term intention a little bit more carefully. So the rational or natural ordering of an act to a specific end is intention. So Aquinas states, to intend is to tend towards something, which is indeed said both the mover and the thing moved. Accordingly, that which is moved to an end by another is said to intend that end. In this way, nature is said to intend an end as it is moved to its end by God, just as an arrow is moved by the archer. And in this way also, brute animals intend an end as much as they are moved by natural instinct towards something. Okay, importantly, intention does not require choosing. The arrow intends to hit his mark. The swallows intend to return to Capistrano every year, and neither of them makes a rational choice to do so. This is not our natural way of thinking about intention, but I think it's a helpful way in the context of twinning, because some acts, such as authorship and creation, can only be intentional. So if a monkey at a typewriter randomly produces the text of Shakespeare's Hamlet, the monkey cannot be legitimately named as the author of that work because the act of striking typewriter keys is not naturally or rationally ordered towards any specific literary end. It's a random act, and a highly improbable one. For all the speculation about monkeys and typewriters, I have yet to see anyone produce a document written by a monkey at a typewriter that actually has any coherent intentionality to it. Similarly, if the growth of a tree randomly produces the image of the Virgin in the bark, the tree cannot be legitimately named as an artist creating this image because the act of growth is not naturally ordered to the production of religious images. Okay, so parenting is also intentional. The terms mother and father most properly refer to those agents who initiate sexual reproductive act as reproductive. Now remember, intention does not have to require choosing. Whether through natural instinct, in the case of animals, through conscious intention in the case of humans, whether out of love of pleasure, or love of each other, or even out of love of violence in the case of rape. Mother and father refer to those agents who act for the end of reproduction, whose biological activities are naturally ordered to that end. So, who act intentionally. Twinning, in contrast, is an unintended outcome of an act that is naturally ordered to development, not reproduction. Consequently, if twinning occurs as an unintended consequence due either to the zygote or to some external agent, it is the father and mother of the original zygote who are the intentional originators of the reproductive act and who are therefore the parents of the embryo. Okay, lastly, ontological identity in form and matter versus, or identity in form and matter versus ontological identity. Does the fact that you're exactly the same as something else make you that thing? So a lot of people have raised this sort of mathematical objection to twinning. The transitive nature of identity requires that if A equals B and A equals C, then B must also equal C. That's basic logic. So if the original zygote is identical to both of the two totipotent daughter cells, then both daughter cells must be ontologically identical. This was an argument made by Burgard. So zygote A is identical to blastomere B, and zygote A is identical to blastomere C, but two things identical with a third thing are also identical to each other, so therefore the identical twins must be the same individual. They must be ontologically identical. So, some modifications of Burgard's concern in light of the facts. First off, twinning is highly unlikely to occur at the two-cell stage, and substantial evidence suggests that in most cases, the first two blastomeres are not exactly identical to each other. So A does not equal B and C. Identical twins have been shown to be epigenetically distinct, so they undergo at very, very early stages, even as early as perhaps the 4-cell stage, specific modifications to the DNA that are distinct in each cell. So if you split at some stage after the 4-cell stage, you're going to have two individuals who are not exactly molecular copies of each other. So consequently, zygote A is unlikely to be identical to both of the daughter cells, although they may be nearly identical. So there are two related responses to the ontological identity argument. So identity or near identity, as is most likely the case, is neither sameness nor ontological identity. And then I'd like to talk about the insights of Kripke on naming and how that impacts this question of ontological identity. So things can be very, very, very close and not the same thing, I think is the take-home point of my argument. So an egg cell and a zygote are more than 99.9% identical in their composition. Egg cells are very, very large, more than 10,000 times bigger than a sperm. So before and after fusion, sperm is contributing a really trivial amount to the molecular composition of this overall entity. And yet an egg cell will live for about 24 hours if nothing happens, and a zygote can live for 70 or more years. Obviously, that tiny little difference is enough of a difference that they are not the same thing. Chimpanzees and humans are more than 98% identical at the genetic level, but I argue that they are distinctive differences between the two of us, despite our near identity. You know, the case of death, in the instant before and the instant after death, a corpse and a living human are 100% identical in terms of their molecular composition, but they're obviously different things. So identity or near identity is not the same as sameness. Paul Kripke also provides an important insight into this problem by drawing a distinction between naming and identity, which is that the attributes by which we fix a name to something does not make that thing be the thing that it is. In more precise terms, the substantial identity of a thing is logically prior to the attributes that distinguish that thing from anything else. So its ontological nature precedes anything we do to call it out as the same or different thing. So the example Kripke gives is this. If we know that Benjamin Franklin is Benjamin Franklin because he was the inventor of the bifocals, and Thomas Jefferson is Thomas Jefferson because he was the author of the Declaration of Independence, and we later find out we made a mistake, that actually Benjamin Franklin did not invent the bifocals, but Thomas Jefferson did. This does not turn Jefferson into Franklin. That's simply because we use an attribute to name something something, or to distinguish one human from another does not make them distinct. Their distinctness is intrinsic to themselves. And that would be true of the twins generated from a zygote that splits. So if we have two totipotent blastomeres, A and B, and even if they are identical to each other, and there's no unique attribute distinguishing them, they're not made ontologically identical by this fact. It may be difficult or impossible for us to fix a name. But in Ford's words, they have separate, concrete existence. And the difficulty of fixing a name is an epistemological problem, not an ontological one. If I was the mother of these guys, I surely would not be able to fix a name to either of them because they just look too close. All right, so what are we going to conclude from twinning? A full and complete, albeit immature human individual arises at sperm-egg fusion. The proximate active potential to develop into a mature human is what makes a zygote a human being. Totipotent blastomeres, if they exist, have only remote active potential for development. Substantial change does not require a corpse. We can split cells amoeba in two and not have to have a dead amoeba in order to make two new ones. Asexual human reproduction, or twinning, is natural until day 14. Parenthood, or the beginning of a child, is an intentional act, and the attributes used to fix a name do not confer ontological identity or sameness to the twins. So, two works relevant to this discussion is the book on twinning, but also the book that I wrote with my brother, who is a philosopher, Samuel, that covers this topic as well. And this is a great resource. I put it up at the beginning, but I'll put it up at the end. So if any of you have questions, I am absolutely happy to have you contact me either after this talk or by email. And I'll do my best to clarify whatever I didn't clarify in the context of this talk. But thank you for your attention. Okay. And if they let me, I'll take some questions.
If you have questions, I will pass the mic to you and you can speak your question, ask your question.
It's a lot of information, so if you'd like to wait, that's why my email's up there.
Better. Do you remember Phil Donahue? He's a Notre Dame grad.
Hello, thank you so much for your talk. My question was a clarification on like the twinning of like the individual. So you're saying if you have zygote A becomes zygote like B and C, is it one person becomes, is that same one person plus a second person and not one person becomes a different and distinct second and third person? Is that what you're saying?
What I'm saying is that the mathematical argument that because the zygote is exactly the same as the two initial blastomeres, each of which can go off and make an identical twin, that therefore all three of those things must be the same thing, right? So there is no actual nature difference between twin A and twin B and the original zygote. I would argue that we don't need to do that. It's not consistent with the data, and that it's not logically required, because my interpretation of twinning is that one of the twins is likely, there's no reason to conclude otherwise, that one of the twins is not the original zygote. And the other twin is newly generated at the time of the splitting, which is likely to be the blastocyst stage.
Thank you so much.
Yeah, sure.
Thank you. Thank you again for coming today. I just had one quick question for you. So you mentioned that twins are epigenetically differentiated as well. I just want to know how early
Yeah.
can we find that out, and what sorts of experiments show this fact?
Yeah, it's a really good question. We know that as early as the 2-cell stage in natural animal development, we don't know this, we know most of what we know about human development only by analogy from animal experimentation. So in developing mammalian animal systems, the first 2 cells that are generated in most cases have distinct characteristics based on patterns of DNA methylation and other patterns of histone modification within the DNA of those first 2 cells. So those so-called epigenetic changes are changes in the protein modifications of the DNA and the proteins with which the DNA is associated. And those differences play out in terms of different contributions of those cells subsequently to the overall developmental pattern. So there's a certain amount of plasticity in that. So that initial difference arises very early. And yet if you challenge those cells by moving them around and putting them in different locations within the embryo, they're able to shift back and forth between those different potential futures. but that doesn't change the fact that they become distinct as early as the 2-cell stage.
First here and then in the back.
Hi. So in in chimerization, what
Yes.
happens is, do the 2 individuals or the 2 organisms that were there before disappear, and then a new organism is made? Is that how you would interpret that?
Well, it's a good question, and I would say it depends on how it is you define. So he's referring to the notion of if you fuse 2 embryos together to make a chimera, right? What happens to each of those original embryos? And I would have to say that both original embryos die, because the newly constituted embryo will have properties that neither of the 2 original embryos would have exhibited had they been allowed to continue their natural developmental course. And I'll just give a simplistic kind of example. If one of the 2 embryos is producing a very large amount of a particular, say, hormone that induces bone growth, but it has very low levels of receptor for that particular hormone, that individual will be average size. If the other embryo is producing relatively low levels of that same hormone, but very high levels of the receptor, that person will be of average size. But if you fuse those 2 embryos together in combination, you'll have an entity that has high levels of hormone, and half of its cells at least will also have high levels of receptor. So that individual will in all likelihood grow to be very, very tall, which neither of the 2 initial embryos would have had as a characteristic for themselves. So novel characteristics will arise as a consequence of the fusion that would not have been present in either of the 2 contributing embryos. So I would say both of those original embryos die. You constitute a new individual with these intermediate characteristics. I mean, another good example would be if you fuse a male and female embryo and you end up with a chimera that's of indeterminate sexual development because of the equal contributions of male and female cells.
Hello, thank you so much for your talk. I had a question about the stage where you said that at twinning, they become like fundamentally different beings.
Mm-hmm.
So I think prior in the talk, you talked about a experiment they did where they injured, I think, an embryo or a zygote, I'm misremembering the words, and like they were able to develop separately. Is the injuring what makes the 2 new beings fundamentally different? And if so, how is, like, if I'm injured, that doesn't, make me a different being? So I think I'm just misremembering, but hope you understand my question.
Yeah. I think it's a tough concept to think about, but the point of the picture I showed of the cartoon where you cut an embryo in half And then that embryo heals itself up, that half embryo heals itself up and continues on in development. That would suggest that the original embryo has been injured, it recovers from that injury, and it carries on with development without even pausing. So in synchrony with uninjured embryos. So I take that as evidence that there's no death, that the original embryo has not ceased to exist, doesn't start over again, and therefore the original embryo persists. Now we can't tell when we split an embryo which one, since each half is going to do exactly that same thing, which one is the original embryo that persists, but one of the halves will have to be a new embryo. And our inability to know which of those two is the original embryo. I mean, there are theoretically ways you could probably determine this on a molecular basis, but not with current technology, because if we had perfect knowledge of the state of the original embryo and its genetic modifications and other things that have happened, we could compare each of the resultant twins, and whoever was closest to the original, we could probably with confidence assign that person as the original person. But the wounding does not cause you to become another person, except in the case of splitting, where one new person has to come out of that split.
Another question here in the back.
Thank you so much for your talk. My question was, can you just talk a little bit about when you have conjoined twins?
Yes.
And like they grow up to be like in like full development and like are born and everything like that. I think my understanding, at least, of like personhood from like a Thomistic standpoint, is that like part of the aspect of it is that you have like a distinct body.
Right.
So how would you like talk about that or deal with that philosophically and biologically?
Yeah, you know, it's a tough one. I think it's things like this, which of course are very rare events, that make it very hard to apply standard philosophical concepts to an actual existent reality. So there are a number of cases. Perhaps some of you might have heard of the Hensel twins. They were born in Minnesota in '91, I think. They're now, you know, young adults. Well, adults. Women who have a single body and two heads. In this case, clearly they have distinct thoughts, memories, preferences, ideas. They're distinct individuals on the level of rationality, but they're a single organism. So how that can exist philosophically, I think that's a tough question. But I don't think it impacts the independent personhood of each of those individuals, despite the fact that they are a shared organism.
First, thank you very much for such an informative talk. And I want to follow up on a talk I heard you give once before that you didn't quite go into. And I remember you're talking there about 5 different ways that identical twins can be created. And you have mirror images, you have a lot of differences. Could you elaborate a little bit more on, what are the differences? Because you do see different kinds of identical twins. And could you?
I'm not sure I would endorse the concept that there are 5 different distinctive ways to get twins. Okay. I think that the exact stage at which twinning is going to occur and the plane of the splitting could give you morphologically distinctive outcomes, right? So if you have a symmetrical splitting along the presumptive anterior-posterior axis of the embryo, you might get mirror image twinning. Whereas if it was more of an oblique splitting, you might not. But I'm not sure that those can really be categorized as ways. So I think you might be fusing this. You might be misremembering the person who proposed 5 different ways of twinning, because I don't recall that I ever did that.
Okay. But you are saying there's different planes of splitting that can in fact produce morphologically somewhat dissimilar individuals with the same
dissimilar kinds of outcomes, makeup. Right? And again, we mostly know this from experimental.
Is there also a question of timing? I mean, just if it takes place at a certain date, versus another? Would we get different, morphologically different twins?
I think perhaps not. I mean, the issue with splitting is I don't believe any identical twinning happens prior to hatching, prior to the escape from the zona pellucida. Because within that tight confining space, all the evidence I presented that the blastomers are just too sticky. They want to adhere to each other. They want to participate in a single unified developmental trajectory. There's no known mechanism whereby you could keep them from doing that if they were in physical contact with each other. So I think that all twinning happens later than the blastocyst stage. And then you have a relatively narrow window after which, between hatching and 14 days, in which twinning can possibly occur.
Okay.
Hi. You mentioned those. This isn't a question. I'm sorry. You mentioned those two women from Minnesota, and I just happened to be from Minnesota and be friends with their neighbor.
Oh, really?
So I have met them, and I wanted to share just like a couple things that like
Yeah.
they've talked about being as conjoined people. This was also over 2 years ago, so may not have it all straight, but they define themselves as two separate organisms, kind of like how a leech attaches to you, and then like you kind of share blood. Like they don't consider each other leeches. They're very friendly
Hopefully
with each
not.
other. But in the way that a leech attached to you shares kind of a bloodstream with you, like they have their own two systems that work for their independent functions. They just happen to have some of
Combined
that system
systems,
shared.
yeah.
I don't know. It was a weird technicality, but I just had
No.
that knowledge that I thought I should share.
But that's how they perceive it. I think, rightfully so. They have not tried to make themselves, a media phenomenon where, a lot is known about their specific biology. But given the nature, or given the mechanisms whereby conjoined twinning happens, and the level at which they're conjoined, they will share the vast majority of their internal functions, as a combined.
Right next door.
Hi, thank you. I found your definition of parenthood as defined by the intentionality of the actors to be very interesting and very powerful. And I was wondering if we could extend that to cases of fertility treatments, in vitro fertilization, artificial insemination. There's many intentional actors that are participating in the creation of this new organism, right? So I was wondering how you would describe the relationships of those other actors to this new being, and what are the human consequences of that relationship?
Yeah, I mean, I think the fact that the answer to that is not simple and becomes quite messy is one of the strongest arguments against doing these kinds of procedures. I think the clearest case would be in a situation where an experimenter working on, say, a donated embryo intentionally splits it. Then he is the parent of both twins. Or at least one of them, the one produced by the consequence of splitting. Less clear to me would be the moral agency of the person facilitating the natural inclination of the gametes to produce a human in the case of IVF, right? Because it's just less clear to me what his role would be. Honestly, I hadn't thought about it, so good thing for me to think about.
Hi, thank you so much for your talk. And forgive my question if it's just my own misunderstanding, but I kind of heard towards the beginning of your talk, you were talking about how legislation, especially the 14-day rule, was kind of the basis of like embryonic research. So since a zygote is, in your definition, like a human being, a human organism, are there ethical problems with embryonic research and research of that nature?
I would say there are huge problems. The reason that we ended up with the 14-day rule was really following the suit of Great Britain that followed the suit of Norman Ford and Clifford Grobstein with this concept of the pre-embryo, But the pre-embryo concept is unsupported by any kind of scientific evidence. It's clear there is an embryo prior to 14 days. And so doing experiment on early stages of human embryonic, early developing human beings would be doing experiments on human beings.
Question over here.
Hello, thank you so much for your talk. I was just kind of curious if the, like, as far as like twinning, like obviously they're like triplets as well.
Yes.
Is there any difference, or how does that process look?
So the great majority of twins, both just twins and then higher order multiples, are fraternal. So more than one egg being ovulated, and each of them being independently fertilized. So they're siblings, but they're not twins in a strict sense, in the sense of the splitting of an embryo to generate them. There are rare cases of higher order identical multiples, right? So identical triplets and identical quadruplets. And given the rarity of those events and the difficulty of producing in the laboratory, we have very, very poor understanding of how that happens. So I can't speak. I could speculate, but I think the speculation would be something along the lines of a splitting originally at hatching, and then due to perhaps some chemical aberration within the embryo itself, a subsequent splitting of either one or both of the twins. But we really have no idea. That's complete speculation. So.
Hello. I was just wondering why, like, the intention to parent is necessary, like, to be a parent, based on the definition that you gave.
Mm-hmm. It's not a natural way of thinking, or a familiar way of thinking about what intention means. But I think if you reflect on the examples I've given you, that Aristotle defines intention as both something possessed by the individual acting on objects and on the object itself. So if I'm an archer and I shoot an arrow and it hits my target, the arrow also intends to hit the target. So he uses this to explain the natural inclinations of living entities. So if a system or an individual or a process is naturally ordered towards a specific end, then you can say that those components intend that end. So the tree intends to, the sapling intends to grow into a tree because it's naturally ordered towards that endpoint. Okay? So in the case of parenting, parenting is something that is a consequence of the natural ordering of the reproductive function of mature individuals. Right? So that's what those body parts are ordered towards. And therefore, you can only become a parent as a consequence of a natural ordering or an intention towards that end. Whereas splitting of an embryo, the processes that are occurring in the embryo that result in splitting are not naturally ordered towards reproduction. They're naturally ordered towards maturation. That's what they're designed to and set up to accomplish. So in the case of a twin that arises because of splitting at the blastocyst stage, the parents intended, the parents' biology was naturally ordered towards the production of a child, and therefore they are the parents. The embryo is not its own parent or the parent of its twin, because the processes of the embryo are not intended towards reproduction.
Maybe just in the interest of time, since it's getting to be almost 8:45, we'll have our final question.
So as sort of an addendum to the third most recent question from the gentleman over there, would you as an expert in the field advocate for the expansion of the legal stipulations regarding research involving human beings to encompass all embryonic research? Would you advocate for the same scrutiny being applied to embryonic research as are applied to any human testing?
Yes.
Thank you.
A very succinct end to a question. Let us now once again thank our speaker for her enlightening talk.